Hello, my name is Cornelius Willacey and I'm a PhD candidate, and I'm going to talk to you about my research project that was conducted at Leiden University about metabolomics development and metabolic phenotyping of mitochondrial Parkinson's disease. This was a part of the SysMedPD project that was funded by the European Union's Horizon 2020 Research and Innovation Grant. The overall aim of the SysMedPD project was to explore and identify novel drug candidates that have the capability of reducing neurodegeneration in patients with Parkinson's disease that is associated with mitochondrial dysfunction. To investigate such a complex neurological disorder requires expertise from several institutes across multiple nations. The Horizon 2020 research program allowed us to bring together academic partners, being Leiden University, where I'm based, Maynooth University, UCL, Lübeck University, and LCSB, based at Luxembourg University. In addition, we were fortunate to have the industrial collaboration with Memetis, the organ-on-a-chip company, and Chondrion, who specialise in therapies for mitochondrial disease. I'm going to give you a little bit of background about Parkinson's disease. It is a neurodegenerative disease that is caused by the loss of dopaminergic neurons within the substantia nigra. In 2015, 6.9 million people worldwide are living with Parkinson's disease, but this number is set to increase to 14.2 million by 2040. This shows that it is a disease that requires active investigation. There is currently no cure, unfortunately, but symptomatic treatments are available. Most people are familiar with the symptoms of Parkinson's disease because they express in a visual fashion, being the resting tremor, bradykinesia, which is the slowness of movement, rigid muscle, and impaired balance. These fall under the category motor symptoms, but there are also non-motor symptoms that are associated with the disease, being depression, constipation, and dysphagia, which is the difficulty swallowing. The absolute cause of Parkinson's disease is not currently understood, but there are risk factors that increase the likelihood of individuals developing Parkinson's disease. The first being age. Over the age of 60 increases the chance. Gender and ethnic factors also take place, so it mainly affects males and those of European descent. There are also environmental factors that have been linked to Parkinson's disease. The first is the pesticide rotenone, the illicit drug MPTP, and it can also be drug-induced by neuroleptics. There are also genetic mutations that are associated with Parkinson's disease: alpha-synuclein, UCHL1, Parkin, PINK1, DJ1, and LRRK2. We chose to focus on the mitochondrial mutations, being Parkin, PINK1, DJ1, and LRRK2 for this project. Mutations in PINK1 and DJ1 have the potential to cause early onset Parkinson's disease. Parkin can cause early onset, but also juvenile Parkinson's disease. LRRK2, on the other hand, sets in with late onset, similar to idiopathic Parkinson's. I've already mentioned the mitochondria quite a lot, and most people are familiar with this organelle that exists within all cells, but I just want to give a quick explanation on the main functionalities. So they're commonly referred to as the powerhouse of the cell, and they're involved in bioenergetic, biosynthetic, and signaling responses. They achieve this by oxidative phosphorylation across the electron transport chains using complex 1 to 5. They also have the ability to mediate cell growth and death and regulate oxidative stress. Now that I have the background out of the way, I want to talk to you about metabolomics, and this was my involvement on the SysMedPD project. Metabolomics is the comprehensive study of the biochemicals that exist within cells, tissues, and biological fluids. To investigate the human metabolome requires consideration of several factors before starting the research. The first is the metabolome coverage. The human metabolome is vast, and you need to make sure that you select the specific metabolites that you think are associated with the disease you are currently studying. The second obstacle is volume-limited samples. This is making sure that you have enough biomass that you can actually detect the chemicals using the analytical systems. we received a range of biological samples. There was microdialysate obtained from mice, in vitro cell samples using induced pluripotent stem cell-derived dopaminergic neurons, urine, and blood plasma. In addition, you also need to make sure you have the most appropriate analytical technique set up to achieve the job. The data obtained by Metabolomics is going to be incorporated into a systems biology model. We also wanted to make sure that all of the data was given in an absolute quantitative fashion. We wanted to know the absolute concentration of each metabolite that we target within our methods. So, to start with, we broke down Parkinson's disease metabolism, and we chose to concentrate on two different areas. Because we're studying the mitochondria, we thought the logical point of view to go to will be the study of the central carbon and energy metabolism, and this is where we investigate the TCA cycle, N-acetylated amino acids, acyl carnitines, and medium and long-chain fatty acids. And then we also have the study of the substantia nigra. So we have the iPSC-derived dopaminergic neurons. So we thought the most appropriate metabolites here to concentrate on would be the neurochemical profile. So this is where we targeted the neurotransmitters, polyamines, and biogenic amines that exist within cells. The central carbon and energy metabolism is very common, but despite this, there are a few quantitative methods that exist out there in metabolomics. So this is where we decided to start from the bottom and work our way up. So we created a derivatization technique using dimethylaminophenacyl bromide to label thiols, amines, and carboxylic acid containing metabolites. In doing so, we were able to capture a broad range of metabolites that can be seen on the right here. In the bottom left shows a chromatogram, so we can capture all of these metabolites within a 10-minute LCMS run in a very cost-effective manner. Additionally, this method was conducted on urine and cells, showing a broad applicability to a range of matrices. Coming back to the volume-limited sample challenge, this was shown on the SysMedPD project. As technologies progress, companies like Mematus are creating three-dimensional microfluidic devices that can culture the iPSC-derived dopaminergic neurons. But with this comes the challenge of having a reduced cell number, so we need to make sure that our analytical methods can keep up with the sensitivity to investigate the metabolome. Here, we were able to quantify 37 metabolites below 10,000 cells, bringing it within the microfluidic range. You can read more about this in the Microchemical Journal publication in the special issue. To capture the neurochemical profile, we utilized the method published by the Robert Kennedy Lab, measuring metabolites using benzyl chloride. The reason we opted for this approach is that benzyl chloride is a very soft derivatization approach, and the chemicals in the tyrosine and catecholamine metabolism, such as dopamine, are very vulnerable to degradation during measurement. We chose to adapt this method by implementing it on a micro-LCMS, which really gave us the ability to increase the sensitivity, so we could also measure the cells that were generated by Mematus. After the implementation of both analytical techniques using DMPA bromide and benzyl chloride, we decided to apply this to the study of the iPSC-derived dopaminergic neurons, looking at the healthy control against the PINK1 mutation. We also had the isogenic control being the PINK1 gene-corrected samples. Here we show the measurement of succinic acid, and we started to see a change in the level of succinic acid between healthy control and PINK1 mutant, and there was no difference between the isogenic control and the healthy control. We also investigated the replacement of glucose with galactose, but this also didn't show a strong phenotype. Therefore, we decided to perturb the cells to see if we could check the resilience. Does the presence of the PINK1 mutation make the cell lines more vulnerable to mitochondrial damage? So we added oligomycin This compound blocks the complex 5 of the electron transport chain, effectively shutting down the mitochondrial function. Here we started to see more of a response in the PINK1 mutant. The level of succinic acid started to reduce, and we didn't see this in the isogenic control. We then tried rotenome, which shuts down the complex 1 of the electron transport chain, but this didn't show the same effect, so we're looking into this and trying to find a reason. With this assay, it's given us the ability to identify mitochondrial dysfunction, and we're also going to incorporate the Chondrian treatments to see if we can identify any new therapies that can be used to treat Parkinson's disease associated with mitochondrial dysfunction. Okay, that brings me to the conclusion. So on this project, we were able to broaden the metabolic coverage by creating a quantitative method that captures essential carbon and energy metabolism. We further optimized existing methods, increasing the sensitivity, making them capable of analyzing volume-limited samples. Using both of these approaches, we now have a novel analytical technique with the ability to identify mitochondrial PD phenotypes, and we created an assay for therapeutic targets. The project SysMedPD has been very challenging but fun, and I've been able to achieve the steps in metabolomics development along with the collaborators that have helped me out on this project. So I'd like to acknowledge Maynooth University, Chondrian, Lübeck University, the colleagues that work with me at Leiden University, Mematus, the LCSB, and University College London. Thank you for taking the time to listen, and I wish you well.